I'll move on to the last part of this webinar, where I'm talking about evaluating protein purity. So to evaluate protein purity, you can use the most common method called polyacrylamide gel electrophoresis. As you can see, you need all these little things to run your gel, and then you will know the size of your protein. You can also do protein quantification using spectrophotometric methods. And in this case, you will have UV visible methods, and you will use like a nanodrop, or you can use Bradford assay. It's a dye, it's a colorimetric method, but you will also need a spectrophotometer to measure that. You can also use another analytical method. This is an analytical chromatography. In this case, it's called reverse phase high pressure liquid chromatography. And you use this other fancy machine. Here you have the detector, the column, the fraction collection, where are you going to pick up your sample. And you need to use a column, but this column, if you see, this column has like a metal outside. Like, because in this case, you are putting a lot of more pressure than the ACTA. It's almost like the ACTA, but this is like more a small detail. The ACTA is more big preparation, but in HPLC, you have really small quantities that you will inject inside the column, and that would be the big difference. So begin talking about page, SDS page. So as you can see here, we have let's say that we have a normal protein that has a good 3D structure, that falls in the right 3D structure. So we have, then we will use SDS, that is an anionic detergent, that will break all these bonds, like the hydrophobic bonds, the hydrogen bonds, the anionic bonds, the ionic bonds, the van der Waals, all these bonds. And then we will have 2-beta-mercaptoethanol that will cut down these disulfide bridges. And this is how our protein will begin to lose shape, and it will begin to be straight. And this SDS page, when it's heated, because we need to heat it up, it will like nicely strain out your protein. And then this part is called sample preparation. And when the protein is straight, we will load it in these wells, and then we will run the gel for the separation, because now all the protein will have negative charge because of the SDS, it's a detergent that has a negative charge. So we will go from the cathode to the anode, and we will separate through the polyacrylamide gel. And then when we reach the bottom, we will stain our gel with coma C blue, so we will identify the protein, and then we will take a picture, and we will have a gel like this. So don't get overwhelmed by my gel, but here I'm showing how you can monitor the purification process with page. In this gel, I have the molecular marker, so you can see here, it's like a balance when you go to the bathroom and you weigh yourself, you know. So you can see here different molecular weights of proteins. And I want you to understand that you can monitor all your purification process using SDS page. So you can begin from the beginning and use a blank, so uninduced control. So this means that you have a bacteria where you didn't induce the production of the protein, and this will be your control. And then here you have a bacteria where you induce the protein So the protein of interest is this size. It's 80 something. So you can see this here because this is uninduced, so it shouldn't have any band. If you will have a band here, you will be worried. But here we have the protein of interest, and here I have different methods to break the cell. So I use sonication, ultrasound, and lysozyme treatment. So, and this protein, the beauty of purifying thermophilic proteins is that you just need to boil them. So this is the sonicated soluble fraction, but then I boil the protein. And when you boil the protein, everything gets degraded but your protein of interest, because this is a special case. This is a thermophilic protein, so you will see it here after boiling it. So I did the same thing for each case. Plus, we also tried the hexanol, because this protein was used for synthesizing in an organic media. So we had to have some hexanol. So if you can see, the protein survived the hexanol and survived the heat shocks, and I think this is the nicest one. This is what I think that's the method of breaking I would choose, because it's nice fat band of pure protein there. And then the gel was 12%. It's like the normal gel that you use for acrylamide. If you have proteins that they are peptides or really little proteins, then you need to have a higher concentration of acrylamide. And the loading was 5 microliters of each in each well of soluble protein. So that's the nice part of using SDS page. So another way to quantify proteins is using UV visible spectrophotometry, where it's a quick and non-destructive method where the aromatic amino acids like tyrosine and tryptophan within the protein will absorb the UV light at 280. So here you see, this is a standard kind of BSA, but this is high concentration. So you can see there is this nice bump here. And this is low concentration, so you have a smaller bump here. And the detection range of this method goes from 0.2 to 2 milligrams per milliliter. If the sample is not pure enough, you can have, like in the crude extract, you will measure all the other compounds, so you will not really measure the real protein content, because there is other nucleic acids or other things in the media that will also absorb at 280. So this is why it's important to do blank of your buffer, especially. So you will blank your buffer so the buffer will not absorb, and then you will get a real concentration of your protein. And everything is based on this law called the law of Beer's law, or Lambert and Beer. So this is the equation that Lambert proposed, and this is the things that we need to get. But this is already calculated by the, in this case, the nanodrop. And then you will get the concentration of based in the absorbance and the extinction coefficient and the path, length path of the. And so this is just a little bit of the background of the equation of what happens when you put your protein in the nanodrop, because the nanodrop will give you already the concentration. But it's good for you to know how this happens, which is the equations that you need to know to do the calculations of the protein concentration. So the other method is rapid colorimetric assays, where you use this dye. This dye has been improved by Biorad. Now it's called Comacy. It was Comacy Blue. Now it's Comacy G250. And this is the dye that will bind in a basic, they will bind to basic and aromatic amino acids. And then, but this dye is in a acidic environment. But when the dye binds to the protein, when the complex is formed, the dye will change to blue. And we can use BSA one more time to do a standard curve. And the detection rate is from 0 to 1 milligram per milliliter. So this is how our standard curve will look like. So this is like a protein that is not high concentration to high concentrated protein. So you will get an absorbance at 595, where you would see that you have a linear part until 1. But after that, it's plateauing, so you don't need to take these 3 points. You just focus in the linear part, do a regression line, and get the data to get an idea how much protein you have in your solution. But the disadvantage with this dye is that sometimes precipitates with detergent. So if you have your protein with detergent, it will precipitate, and also sometimes some of the proteins they won't be soluble at low pH, so then it will be hard to measure them. So then you need to choose another colorimetric method that suits your protein of interest to be more to find a reliable method to measure or quantify the protein. So this is the last method. It's called protein quantification using reverse phase HPLC. So if you can see here, this technique can separate big proteins and small proteins like peptides. And everything is based in the hydrophobicity because the support is silica that is non-polar. And it has alkyl chains. The most these alkyl chains, they give the hydrophobic character to the silica and the method. So here I'll tell you how it works. So you have to go from a hydrophilic solvent to a hydrophobic solvent in a gradient. So you need to mix, and then your proteins, they will go first inside the silica and get in contact with these alkyl ligands, and then you will see that the first proteins that they will get eluted, they will be the hydrophilic molecules, and the hydrophobic, they will stay, and then you will change solvent to more hydrophobic solvent, and then at the end the hydrophobic proteins will go out. And that's the beauty of this method. So here we have an example where we are using a monoclonal antibody called Campat. And in this case, the alkyl chains, usually they are C8 or C18, but this is a C4 column. And the protein, the antibody size is 150 kilodaltons. So what they did, they did a gradient of acetonitrile and water, and you go from more water and then to more acetonitrile, 70%. And you can see that this method is really, okay, they detected they quantify the protein at 214 nanometers, and the method, the protein was eluted at 9 minutes. So this method is really fast, and the separation is so good because this is a really, really pure antibody, and you can later on use this peak and connect the HPLC to a mass spec to really see each part of the amino acids of the antibody. So this method is fast, so you can, if you work in companies, you need to know that your product that could be a vaccine needs to be super pure. So you need to get this method as part of your analytical process of evaluation of purity. As well, here you get the size besides the purity, so it's like a combination of SDS page and the quantification, because you will quantify and you will also see the size of your protein. So the last tips for this session will be SDS page is a useful method to monitor the presence of your protein of interest during the whole purification process, especially if it's the first time that one is attempting the downstream processing. Success in quantifying the total amount of pure protein relies on removing impurities from the sample. When using colorimetric methods for protein quantification, choose a method that is compatible with the physicochemical properties of your target protein. When choosing a buffer for quantification method, be sure that it doesn't contain reactive components that they could absorb wavelength at the same range of your amino acids. So we will avoid overestimation of the actual concentration. So here I put you 4 links. They are really good if you want to really understand more in detail all the things I talked to you today. You can go into these 4 links and check them out. I highly recommend you to do that. And then questions.
So we have a couple here. The first one has to do with how you're dealing with your different protein elutants. So should beta-mercaptoethanol be added while doing SDS page, even if the protein does not have disulfide bonds?
Yes, they do by default. And now, because we get a lot of material, before we used to prepare the sample buffer, but now we just buy it. And you mix them and then you do it, yeah, you use it. You can, it won't harm having them. The beta-mercaptoethanol, of course, if there's no disulfide bonds, maybe you won't interact with anything, but yes.
All right, and the next question we have is, how do we choose the method to quantify the proteins? Is it dependent on the type of protein or other parameters?
Yes, so I can say that it depends on okay, it depends on many things. Like it could be, you will need to use whatever you have in your lab. So if it's a spectrophotometer, you have to choose the spectrophotometer. But if you have, I mean, if you want to use a colorimetric method, you can use a colorimetric method. The most important thing is that you need to be sure that you take in account the physicochemical parameters of your protein. Like for example, if my protein is insoluble at low pH, I cannot use Bradford, then I need to use another method like BCA if I want to use colorimetry. So then I use BCA. That will be another way of quantifying the protein. So it would be, I will always prioritize the physicochemical parameters of the protein, and after that, what I have in the lab. If I have access to a HPLC, yeah, I would like to use that. But maybe you don't need to have a HPLC. You can have a spectrophotometer in your lab, and then you can use that. Yes.
One thing just to keep in mind with Bradford is it is technically measuring specific amino acid residues that are more basic. So if you have a protein that doesn't have a lot of those in there, it may bias your results when you're trying to determine what your concentration is. So that's another thing where Tanya was talking earlier about knowing what your sequence is. This is where it can become very important to kind of know that information ahead of time if you can get it.
Yes, yes. There is some proteins that they don't even have like aromatic amino acids. So it's really important to get into the sequence of your protein.
Great. So we have another question here, and we've already had somebody kind of follow up on it. Some proteins exist as heterodimers or trimers in native states. How do we purify such proteins?
You just purify them as like with the same methods that I showed. You don't do other modifications. Like if you purify them in the correct way, trying to keep the 3D structure and all based on all the physicochemical properties, if they are dimers or tetramers and they need to work together as a dimer, in your pure solution, they will bind and they will find a way to work. So you don't have a special way to separate them. You use the same chromatography methods to separate them.
One of the comments off of that was that you could use a his tag to kind of pull those down out of solution as you're working on them, so then you can study them more in particular.
Yeah. Also, you could do that, yes, because the his tag can bind proteins in any conformation. Yes.
Okay, that's all the questions we have so far. With this guy. So any other questions? We'll do a last call. All right, not seeing anything yet. Maybe give this another second. All right, well, thank you everyone.
Yes, thanks for listening, and I hope that this set of webinars that we put together, I hope they were really informative and helpful for all the iGEMers. I hope the experiments are going good, and yeah, I think that would be all, right?